Hello and welcome to this Bio43 pre-lab video. Today we're going to talk about ANOVA. This is the statistical test to use when you have a categorical independent variable and a continuous dependent variable, and you have 3 or more treatments. We'll also talk about controlling confounding variables, which will help when you're thoughtfully designing your own experiments. In the previous video, we addressed a situation where we compared the size of tetrahymena grown in the lab to wild tetrahymena, hypothesizing that, because growth media has a higher concentration of nutrients than lake water, lab-grown tetrahymena are larger than tetrahymena found in nature. We ran an experiment which compared the length of tetrahymena in the lab to tetrahymena in fake lake, and indeed saw a statistically significant difference. However, I suggested at the end of that video that confounding variables make it very difficult to say whether the mechanism in our hypothesis was really supported. The nutrient composition is not the only difference between the lab sample and the lake sample. The tetrahymena at the 2 locations have different genomes. The 2 populations are exposed to different temperatures. The lake tetrahymena might need to invest more energy into evading predators, and therefore have less resources to put into growth. Any of those factors could be responsible for the difference in size we observed last time. But we could design a better experiment that controls for these variables. In addition to the 2 treatments we previously discussed, we'll be adding a 3rd treatment. Take some lake water, sterilize it, inoculate it with some tetrahymena from the lab, and grow it in the same incubator as our lab stocks. Since the tetrahymena originate from the lab stocks, the DNA is the same. Since they're grown in the same incubator, there's no difference in temperature. And since they're in sterilized lake water, there are no other organisms to compete with. Each of these confounding variables is now controlled for. Let's adjust our prediction to account for the new experimental design. Obviously, we still predict that lab tetrahymena in lab media will have greater average length than tetrahymena from fake lake. Now we would also predict that lab tetrahymena in lab media will be larger than lab tetrahymena grown in sterile lake water. Finally, we would expect little to no difference between the lake tetrahymena and the lab tetrahymena grown in lake water. The differences between these treatments may still have some effect, but the nutrient composition is the same. So if our hypothesis is correct, the difference between these controls will not be as great as the difference between either control and the experimental treatment. Now let's look at the data with this new treatment included. Again, check the integrity of the data, get our summary statistics, plot the data. All of this is exactly as demonstrated in previous videos, so let's hurry through that. You can see that the lab-grown tetrahymena are larger than the ones from fake lake, as we saw before. And the lab tetrahymena grown in lake water are somewhere in between, but appear closer in length to the tetrahymena from the lake. We'll again need statistics to tell us which differences are significant. However, the t-test can only compare 2 treatments. You could run 3 t-tests to look at each pair of treatments, but that gets tedious, and becomes impractical once you need to start comparing 4 or more treatments. There is a quicker and more robust option in ANOVA. For that, we're going back to Vassar-Stats. Click on ANOVA this time, just the basic one-way ANOVA. Scroll past these directions. Now, one confusing aspect of Vassar-Stats is that when they ask for the number of samples, they really mean the number of treatments or conditions. So our sample size is 10, but we input 3 here, the number of treatments we're analyzing. Independent samples, weighted, then just copy and paste our data into the 3 sample columns here, and click Calculate. Again, we have a data summary. The n values are all 10. The means match the Excel values. Looks like we transferred the data properly. The second table has the key stat values you'll want to record for the ANOVA analysis. Instead of a t statistic, we record an f statistic here. There are actually 2 degrees of freedom to record here. One connected to the number of treatments, and one connected to the number of samples. And finally, here's our p-value. Our value is about 0.003, below our threshold of 0.05. So there is indeed a statistically significant difference between the groups. However, this doesn't tell us which groups are significantly different from each other. For that, we need a secondary analysis called 2-key HSD, which conveniently, Vasterstats has already performed and displayed the results in the table below. Note that 2-key HSD will only be performed if the overall ANOVA p-value is less than 0.05. If there's no overall difference between the groups, there's no need to compare group by group. Vasterstats just uses the M1, M2, M3 notation to describe each treatment, so you have to remember the order you input them. The only values you need to record are the p-values for each pair. In this case, there is a significant difference between the lab-grown tetrahymena and those from fake lake. P is less than 0.01. There is no significant difference between the lake tetrahymena and the lab tetrahymena grown in lake water. But there is a significant difference between the lab tetrahymena grown in the lab media and those grown in lake water. P is less than 0.05. So we once again reject our null hypothesis and support the alternative hypothesis that lab-grown tetrahymena are larger than tetrahymena found in nature because lab growth media has more nutrients than lake water. And because of how we set up the experiment, we have good support for that mechanism. But keep in mind, this is still just support and not proof. We never really prove anything in science, but as we perform more and more experiments and gather more and more support, we can reach the point where a given hypothesis becomes the only reasonable explanation. That's it for this video. I hope you now feel confident using ANOVA in situations where you have a continuous dependent variable and categorical independent variable with 3 or more treatments. You should also now have a better understanding of confounding variables, which will help you when you're designing your own experiments. See you next time!